Welcome to today's lesson on how genetic variants in both non-coding and coding DNA can influence an organism's phenotype. In our roadmap for today, we will explore how genetic variants in the non-coding DNA of a gene can impact phenotype by influencing the binding of RNA polymerase, which in turn alters the quantity of protein produced. Additionally, we will explore how genetic variants in the coding DNA of a gene can affect phenotype by changing the sequence of amino acids, thereby altering the activity and function of the resulting protein. So far, you have learned about the structure of DNA and the process of transcription. This foundational understanding is essential as it allows us to explore the more nuanced ways in which variations in the DNA sequence can lead to differences in the physical traits and functionalities of organisms. Knowledge of how changes to the DNA sequence can affect protein production is crucial for fields such as genetics, medicine, and evolutionary biology, as it helps us comprehend how variations can lead to diversity, disease, and adaptation. Let's get started. Non-coding DNA may not produce proteins directly, but it plays an essential role in regulating gene expression. Here's how mutations in non-coding DNA can influence phenotype. Mutations are random changes in DNA sequence. When these occur in non-coding regions, they create genetic variants. These mutations can affect how well RNA polymerase binds to the DNA. If RNA polymerase binds less effectively, the transcription process becomes less efficient. Less efficient binding means less mRNA is produced, leading to a decrease in the amount of protein synthesized. It can further impact on phenotype. Since proteins carry out various functions within an organism, a decrease in their production can lead to phenotypic changes. For example, if the protein in question is an enzyme, its reduced levels can alter metabolic pathways or other cellular processes. Imagine a mutation in the non-coding region of DNA that affects the production of a protein necessary for pigment production in the fur of an animal. If RNA polymerase cannot bind effectively, less pigment protein is made, possibly resulting in a lighter colored fur or albinism. Now, let's explore how mutations in coding DNA can affect phenotype. The coding region of a gene directly specifies the amino acid sequence of a protein. Here's a step-by-step look. Mutations in the coding region create genetic variants, leading to a change in the sequence of mRNA after transcription that can alter the amino acid sequence of a protein during translation. Since the sequence of amino acids determines a protein's structure and function, any change might lead to a different protein shape or function. These changes can have various effects. The protein's activity might increase, decrease, or stop altogether. The altered protein function can lead to noticeable changes in an organism's traits. For example, a change in an enzyme's activity could affect a metabolic pathway, leading to a disease or altered physical traits. Consider a mutation in the coding region of a gene responsible for creating hemoglobin, the protein that carries oxygen in the blood. A single amino acid change can lead to sickle cell disease, a serious condition where the red blood cells become misshapen and less efficient at transporting oxygen, thereby affecting the organism's overall health. By understanding how genetic variants in both non-coding and coding DNA affect phenotype, we gain deeper insight into the complexity of genetic expression and variability among organisms. These concepts are crucial for fields like genetics, medicine, and evolutionary biology. Now let's work through some questions to consolidate our knowledge on how genetic variants can affect phenotype. Question 1. Describe how a change in the sequence of coding DNA affects the protein synthesis process. We know a change or mutation in the DNA sequence will alter the sequence of bases. The altered DNA sequence may result in a different mRNA sequence during transcription. A change in the mRNA may lead to the incorporation of incorrect amino acids during translation, potentially resulting in a non-functional protein or a protein with a different function. This can affect the phenotype of the organism. In this next question, we must compare the effects of genetic variants in the non-coding versus coding regions on phenotype. Firstly, remember that non-coding regions influence transcription levels, while coding regions determine protein structure. Variants in non-coding regions affect RNA polymerase binding, therefore altering the amount of protein produced, whereas variants in coding DNA change the amino acid sequence, directly impacting the protein's properties. In today's lesson, we explored how genetic variants in both non-coding and coding regions of DNA affect phenotype. We began by discussing DNA structure and its role in inheritance and evolution. Our first was on non-coding DNA, which, though it doesn't code for proteins, regulates gene expression. Variants here can influence protein production by affecting the binding of RNA polymerase, affecting traits. We then looked at coding DNA, where genetic variants can alter the amino acid sequence of proteins, impacting their structure and function, and thus, the organism's phenotype. This lesson highlighted the complex relationship between genotype and phenotype, providing a foundation for understanding genetic mutations and their effects in various fields.